A fish that sees through its skin. Odins, like many marine creatures that inhabit coral reefs, are experts at changing the color of their skin. New research has shown that these fish can see themselves through their skin, which has a built-in light-sensing mechanism independent of the eyes. Moreover, it turns out that they don't even need to live to maintain this ability. A few years ago, during a fishing trip in the Florida Keys, biologist Laurie Schweikert caught a wild boar, Lachnolimus maximus. It is a common species found in the western Atlantic from the coast of the United States to northern Brazil. Inhabits areas near coral reefs. It reaches up to 1 meter in length and up to 7 kilograms in weight. He is known for his incredible camouflage abilities. Representatives of this species can change color in a few milliseconds to blend in with the environment. Schweikert, after catching the Odin, threw it on the deck. After a while, she noticed that the fish had adjusted its coloration to the color of the deck boards. It wouldn't be surprising if it weren't for the fact that the fish was already dead. This made her wonder if this species could detect light with its skin, independent of its eyes and without the involvement of its brain. The researcher began researching the species. In 2018, together with Somke Johnson from Duke University, she showed that fish possess a gene encoding opsins, light-sensitive proteins. They occur e.g. in the retina and are involved in the vision process. Only in Odians opsins are activated in the skin, and this gene is different from the genes that encode the opsins found in their eyes. Studies have also noted that other color-changing animals, from octopuses to geckos, also produce light-sensing opsins in their skin. But exactly how they use them to change color has not been clear. When we found the gene in Odins, I looked at Somke and asked, "Why have a light detector in the skin?" Said Schweikert, now an assistant professor at the University of North Carolina at Wilmington. One hypothesis is that light-sensing skin helps animals learn about their surroundings and camouflage better. However, new findings, published in the journal Nature Communications, suggest a different possibility. These fish may use opsins to look at each other. In the new analysis, the researchers took pieces of skin from various parts of the fish's body and photographed them under a microscope. Up close, the Odin skin looks like an example of pointillism. a painting technique in which the composition of the painting is built through densely strewn, multicolored points. The same is true of the skin of these fish. Each colored dot is a specialized cell called a chromatophore containing granules of pigment that can be red, yellow or black. The movement of these granules is responsible for the change in skin color. As the granules spread across the cell, the color appears darker. When they focus on one tight space, the cell becomes more transparent. The researchers then used a technique called immunotagging to locate the opsin proteins in the skin. They found that in odins, opsins are not produced in color-changing chromatophore cells. Instead, they are in other cells directly below them. Images taken with a transmission electron microscope revealed a previously unknown cell type, just below the chromatophores, filled with the opsin protein. This means that light hitting the skin must first pass through the pigment-filled chromatophores before reaching the photosensitive layer. Scientists estimate that the opsin molecules in Odin's skin are the most sensitive to blue light. This happens to be the wavelength of light that is best absorbed by the pigment granules in the chromatophores of the fish. The findings suggest that the fish's light-sensitive opsins behave somewhat like the inner polaroid membrane. These animals can literally take a picture of their own skin from the inside out. In a way, they can find out what their skin looks like, Johnson said. To be clear, we're not saying Odin's skin works like an eye. We don't have any evidence to suggest that's what's happening in their skin, Schweikert said. Because the eyes not only detect light, but also transmit stimuli to the brain, from which images are created. In the case of Odians, the researchers point out, we are dealing with a sensory feedback system that allows the fish to monitor its own skin as it changes color and adjust it to what it sees with its eyes. These fish seem to be watching their own color change, Schweikert explained. The study authors say the work could pave the way for improved devices such as robotic limbs and autonomous cars that need to adapt their behavior without relying solely on camera feeds. The sensory feedback system is one of the tricks that technology is still trying to master. This study provides a good analysis of a new type of feedback system, Johnson said. If you didn't have a mirror and couldn't bend your neck, how would you know if you're dressed appropriately? It may not matter to us. But for creatures that use their color-changing abilities to hide from predators, warn off rivals, or woo mates, it can be life or death. SZWH Kurt said. Scientists have fully sequenced the Y chromosome. Due to its complexity, sequencing the Y chromosome has been a major challenge for scientists for decades. But finally it worked. Telomere to telomere, T2T, researchers have published the first complete Y chromosome sequence. Scientists in the telomere to telomere, T2T, consortium published the full sequence of the Y chromosome in 2 papers published in the journal Nature. This work fills gaps in the human genome. 20 years after it was published its first version. They also show a complete catalogue of male sex chromosome genes and how they are organized. The first paper detailed the arrangement of the repeating regions and identified dozens of new genes. The second shows that this arrangement, as well as the number of genes, varies greatly from person to person. Complete sequencing of the Y chromosome was once considered an impossible task. This chromosome is full of repeated and inverted sections of DNA, making it difficult to reassemble these pieces in the correct order. But using state-of-the-art methods, we finally made it. In doing so, researchers have taken the final step in the long-awaited full sequencing of the human genome. The first publications on reading the human genome appeared over 20 years ago. It was a huge achievement, and the researchers of the Human Genome Project were behind it. The deciphered genome covered most of the protein coding regions, but about 8% of the genome was missing to be sequenced. It took 2 more decades to fully understand the human genome. The difficulties were mainly due to the fact that the missing 8%. They are repetitive pieces of DNA that were difficult to match up with the rest. The complete sequence could facilitate the detection of gene variants on the Y chromosome and link them to specific traits, as well as the development of personalized treatments for sex-specific genetic diseases. The findings also show that the Y chromosome has evolved and shows significant differences in size and structure between individuals. The Y chromosome is one of the 2 chromosomes, along with the X chromosome, that determine a person's biological sex. Females typically have 2 X chromosomes, while males typically have 1 of each type. The Y chromosome is important for the development of male sex characteristics. contains a sex-determining gene that encodes a protein that promotes testicular development and blocks the development of female organs such as the uterus and fallopian tubes. Scientists believe that the Y and X chromosomes were once identical. Over time, the gene-containing portion of Y has shrunk to 1/6 the size of that region in X, and now contains half as many genes as its larger counterpart. Some researchers have argued that this degeneration may progress gradually, possibly leading to complete loss of the Y chromosome, as has been the case with some animal lineages. Using a combination of state-of-the-art DNA sequencing and machine learning technologies, as well as knowledge gained from more than 20 years of efforts to understand the human genome, Adam Phillippi's team succeeded in completely assembling the Y chromosome. Sequencing machines and genome assembly software could not penetrate areas where the DNA sequence contained highly repetitive stretches. Repetitions could easily be missed or incorrectly connected, which accumulated errors. As sequencing technology advanced and costs fell, scientists reduced the number of gaps or incorrectly assembled sequences, culminating in the publication in 2017 of a reference genome called GRCH38. They became the basis of work and reference for many, but still contained gaps. In new work, scientists have discovered that the Y chromosome contains 62,460,029 DNA base pairs of letters that make up the rungs of the DNA ladder. This is 30 million more than the GRCH38 reference genome, which has been continuously refined over the past 20 years. The team also corrected errors in GRCH38 and discovered the complete structures of several gene families involved in sperm production. It also identified 41 new protein coding genes and gained insight into non-coding regions called satellite DNA, which do not contain protein blueprints, as well as sequences that in the past were often mistaken for bacterial DNA. In the course of their research, the team also developed special software called VACO, thanks to which the Y chromosome was correctly assembled. But it concerned only one donor, and this chromosome was called T2TY. In a separate study, researchers used VACO to fully assemble the Y chromosomes of 43 men from populations in Africa, the Americas, Asia and Europe, who donated DNA samples to the 1000 Genomes Project, an international project to create a catalogue of human genetic variation. These sequences reflected the degree of modern Y chromosome diversity, with the 43 chromosomes and T2-TY sharing a common ancestor around 183,000 years ago. The sequences ranged from 45.2 million to 84.9 million base pairs in length and, taken together, show that the Y chromosome diversified significantly in size and structure during this time. More discoveries on the Nazca Plateau. 168 hitherto unknown geoglyphs have been found. A team of Japanese archaeologists, in collaboration with their Peruvian colleagues, have discovered 168 hitherto unknown geoglyphs in the Nazca Plateau in Peru. Newly discovered terrestrial drawings show, among others, people, birds, killer whales, camels or cats. The geoglyphs were discovered during field research, which lasted from June 2019 to February 2020. Drones and artificial intelligence helped in the discoveries. Huge geometric figures, likenesses of monkeys, fish, spiders and other animals up to hundreds of meters long, are visible in all their glory only after rising into the air. There are hundreds of them on the Nazca Plateau. Located in the desert in southwestern Peru, these huge terrestrial drawings are the world's most famous example of geoglyphs. Scientists believe they are up to 2,500 years old in some cases, though some researchers believe they may be much older. The newly discovered terrestrial drawings were found by archaeologists at Japan's Yamagata University. Scholars believe that the drawings date from 100 BCE by 300 CE. As many as 36 of these geoglyphs were discovered in the area of Aja, near the city of Nazca. Earlier, in 2014 and 2015, the same research team discovered 41 geoglyphs in the region, which led to the creation of an archaeological park in the area in 2017 in cooperation with the Peruvian Ministry of Culture to protect them. The Nazca Desert in Peru, between the cities of Nazca and Palpa, is like an art gallery. However, in order to see the works well, one must take to the air lines. Images of animals or geometric patterns in the desert in southwestern Peru have been a source of fascination for decades. They were created by the Nazca Indians by removing the top layer of red desert stones to reveal the pale layer beneath them. Geoglyphs stretch across a vast area of the desert and there are hundreds of them, although researchers say that only a small part of the collection has been known. One of the research teams working on the site are archaeologists from Yamagata University. Japanese scientists have already achieved great achievements. 3 years ago, they reported the discovery of over 140 unknown geoglyphs. Interestingly, artificial intelligence helped them in their discoveries. Japanese scientists are collaborating with local scientists. The joint research led to further discoveries. Using aerial images taken by drones, the researchers identified 168 new geoglyphs. About 50 of these large-scale drawings feature human-like figures. Other geoglyphs carved into the ground depict birds, killer whales, cats and snakes. A few of them are plain, straight lines, perhaps unfinished projects. It is difficult to say when the drawings were made, but fragments of clay vessels that have been found near the geoglyphs date from between 100 BCE and 300 CE. Many of the drawings were made on flat terrain, which makes it difficult to see them from the nearby, low hills. The drawings were created by removing rocks and earth to reveal a contrasting colored soil, but erosion has rendered them invisible. However, field research using high-resolution aerial and drone imagery revealed hitherto unknown geoglyphs. As with previous studies, the researchers used artificial intelligence algorithms that can detect different patterns faster and more reliably than the human eye. The Nazca lines are one of history's most intriguing mysteries. And although from year to year researchers find new drawings, this does not bring us any closer to understanding their function. For some unexplained reason, the communities inhabiting the territory that now belongs to Peru created straight lines, various shapes and geometric figures, as well as figures of animals and people, which can only be admired in all their glory from above. Over the decades, terrestrial drawings have been interpreted by scientists in different ways. Geoglyphs were discovered in 1926. Initially, they were taken for irrigation systems, then for the sacred paths of a religious nature, but modern science really does not know for what purpose they were created. There are several concepts. Some scholars believe that these are systems of trade routes. Others say that these are types of fields where sports competitions were played. There are also concepts that say that they are an ornament that was supposed to please the eyes of the gods. Others, in turn, emphasize their connection with astronomical observations, because some of the geoglyphs reflect constellations. However, the leading thesis is the one proposed by the German researcher Marie Riker. According to her, the Nazca drawings were used to determine the times of sowing and harvesting. Some of them indicate the points of sunrise and sunset during the autumn and spring equinoxes and the summer solstice.